So students, now we are just going to start with a new topic. In the previous topic we have seen the development of embryo or embryogeny. Actually we are studying the post fertilization events in an angiospermic plant. So in a post fertilization event we have observed the four different processes. First one is the development of endosperm. It is the first and very most important process because endosperm is going to provide the nutrition for embryogeny. So first of all the endosperm is developed, and afterward the development of endosperm, what happens, the zygote is going to convert in form of the embryo. So that is what embryogeny we have seen, the development of monocot embryo and the dicot embryo. Now it's time to see the third step, and finally we'll see the fourth step. So third step is the formation of seed, how embryo is going to convert in form of the seed, and the finally how the ovary is going to convert in form of the fruit. So four different steps are there which are being completed after the formation, after the completion of the fertilization. So let us see the third step after completion of the two step: formation of endosperm, development of embryo, and now the development of seed. And listen my dear student, here we are discussing both the topic together, the development or evolution of the seed, or development of seed, origin of seed, or the structure of seed. Both the topics are the similar. So here we are just going to share the common information regarding that. So before starting the seed, we must understand what is the meaning of seed. So students, the seed is considered as an fertilized, ripened, mature ovule. You know very well, the ovule is the structure which is undergone in the process of fertilization, because here the embryo sac is located, and inside the embryo sac the egg apparatus, egg cell, the gamete cells is present there. So fertilization is going to be taken place there, and finally this ovule is going to convert in form of the seed. So this is an important question. Whenever the question is there, which structure is going to convert in form of the seed, so you must take care of that. The ovule is the structure which is going to convert in form of the seed. So ovule, when it gets mature, so it will form a seed. When it get fertilized, it forms the seed. It get ripened, that time it is going to form a seed. So now it is clear to you all what is seed. Seed is considered as the fertilized, ripened, and the matured ovule. So is it clear to you all what is seed? Seed is the fertilized, ripened, and mature ovule. One very important concept about the seed is that seed is considered as an inactive embryo. Seed is considered as an inactive embryo. What is the meaning of inactive embryo? Just try to understand what is the meaning of inactive embryo. My dear students, normally the activity of embryo is considered as the germination, capacity of germination of the embryo. Whenever the embryo is getting active, whenever the embryo is active, so embryo is going to germinate itself and forms the new plant. So here the seed, you can observe very well, the seed can remain for a longer time without germinating itself. So that is why the seed is considered as an inactive embryo. Are you getting my point? Seed may remain for a longer time without germinating itself, but seed is itself an embryo. So seed is considered as an inactive embryo. Whenever the seed is getting the favorable condition for the process of germination, for the requirement of germination, whatever the conditions are required for the germination, whenever the seed is getting that conditions, the seed is going to germinate itself. That time the inactive embryo is getting activated for the purpose of germination. So this point is clear to you all. Seed is what? Seed is actually the fertilized, ripened, mature ovule. Along with that, seed is an inactive embryo. So both the points are clear to you all. Now very important question that I am just going to discuss here. Normally there in the NEET examination, the competitive examination, the question is asked which process is going to begin, or by which process the seed formation is going to start, which process is providing a kind of stimulus for the process of seed formation. So remember, the double fertilization is the process which triggers the transformation of ovule into the seed. So double fertilization is the process which triggers the transformation of ovule into the seed. So is it clear to you all which process is responsible for the formation of seed from the ovule? So the process that is double fertilization is triggering the process of transformation of ovule into the seeds. What is the meaning of transformation of ovule? So ovule is the fertilized ovule is the fertilized ovule is the mature or fertilized ovule, so that is going to convert in form of the seed. So that is what, how it is being converted after the double fertilization only, because double fertilization is going to provide the nutrition for the purpose, and the nutrition is being given in form of the endosperm. And you knows very well, during the double fertilization the triple fusion occur and the PEN is being produced. So due to that PEN, what happens, the endosperm is going to produce, and that endosperm formation will start the embryogeny, and as well as it will start, so as well as the embryogeny start, it is going to start finally the process of seed formation or conversion of ovules into the seeds. So now after this, the basic concept, now we are just going to understand what are the changes, or what are the process, or what are the mechanisms, what are the basic structures of the embryo sac is going to convert in form of the seed. So whenever the seed is going to form, so what changes embryo sac has undergone. So we are just going to see what are the changes that an embryo sac has undergone after the formation of seed. So let us see the change number 1. The zygote is there. The zygote is going to convert in form of embryo. We knows very well the first change that is being taking place. So after fertilization, in the previous lecture only we have seen the zygote is undergoing in the process after absorbing the nutrition from there. So zygote is going to form the embryo by the embryogeny. The next change you knows very well, in the previous lecture only we have seen the double fertilization is the process by which the PEN is going to produce, and that PEN of the embryo sac is going to form what? Endosperm. So first change is zygote is going to form the embryo, and the second change is what? The PEN is going to produce the endosperm. The endosperm is usually means to provide, or is usually produced to provide the energy during the embryogenesis. So normally the endosperm is being consumed during the embryogenesis, but in some of the cases, what happens, the endosperm may remain persistent. Some of the cases endosperm may get consumed thereon. So on the basis of the presence or absence of the endosperm, the seeds may be of the following type. On the basis of presence or absence of the endosperm, the seed may be of the following type. So on the basis of presence or absence of endosperm, seeds are of two types. Basically the first one is considered as the albuminous seeds. Albuminous seeds means the seed which bears or which has a persistent endosperm. So due to the presence of endosperm always, that seed is also called as endospermic seed. Are you getting my point? The albuminous seed is a seed with a persistent endosperm, where the endosperm is present even after the embryogenesis is being completed. Are you getting my point? What is albuminous seed? Albuminous seed is the seed where endosperm is persistent even after the embryogenesis is being completed. So because of presence of endosperm throughout the life of the seed, it is called as endospermic seed also. So endospermic seed is always having endosperm, that is why this is called as albuminous and the endospermic seed. Then what is the importance of that endosperm present in the seed or albuminous seed? So this endosperm will be utilized during the germination of the seed. So what is the importance of that? That endosperm is utilized during the germination of the seed. So let us take the example, let us see the example of the albuminous seeds. Remember my dear students, in monocot seeds, whatever the portion you are using for the purpose of your nutrition, for the purpose of your energy, that portion you are eating the endosperm of that plant. Are you getting my point? Whatever the portion of the seed you are taking for the purpose of nutrition in monocot plant, that is actually the endosperm. So here the albuminous seed is endospermic seed, means they are having the persistent endosperm. So example of the endospermic seed is basically the monocots are there. Most of the monocots are having the albuminous seed or the endospermic seed. Like that you can see, if you are just using maize, means you are eating the endosperm of the plant. If you are using wheat, you are eating the endosperm of the plant. Rice, barley, coconut, whatever the monocot seeds are there, that seeds are used as the we are using the endosperm of those seeds for the purpose of nutrition. Along with the monocots, some of the dicots are also reported with the endospermic seeds are albuminous seeds. So what are those dicots? Like that castor, papaya and cotton. Castor, papaya, cottons are the dicots which are being considered as, which are being reported with the persistent endosperm, or they are having the albuminous seeds. So this is clear. The albuminous seed is the seed which is having the persistent endosperm, which is not being consumed during the embryonic life of the seed. Now the next one, based on the presence or absence of the endosperm, X-albuminous seeds. X-albuminous seed is the vice versa of the previous one. It is the vice versa of the albuminous seed. X-albuminous means to say the seed where endosperm is consumed during the embryogeny. What is X-albuminous seed? The endosperm which consumed, the seeds in which endosperm is consumed during the embryogeny, and this is why this is called as what? Non-endospermic seeds. This is what called as non-endospermic seed. So is it clear to you? X-albuminous means what? Non-endospermic seeds. Why this is called as non-endospermic seeds? Because the endosperm is being consumed during the embryonic development. So what is the then how they are maintaining their prolonged life, the maintenance of the seed till the time of germination, and from where do they get the energy for the germination, nutrition for the germination? As in case of albuminous seed, we have seen that endosperm is itself providing the energy, because endosperm is the source of stored material. So here in case of X-albuminous seed, the stored material is present in form of the cotyledons. Are you getting my point? Here in case of X-albuminous seeds are non-endospermic seeds, because the endosperm is consumed. So stored material is present in form of the cotyledons. So is it clear to you all? And most of the dicot plants are having X-albuminous in nature. Most of the dicot seeds are having non-endospermic in the nature. The example for dicots like capsula, gram, pea, beans, groundnut, etc. But here also, as the previous one, there in case of the albuminous seeds, some dicots are also reported with the albuminous seeds. So here also some of the monocots are there, those who are being reported with the X-albuminous seeds. What are those? Orchids, money plants, Vallisneria. These are the plants, those are monocots and having the X-albuminous seeds, means their endosperm is being consumed during the embryonic development. Now third category of the seeds presents are on the basis of presence and absence of the endosperm. Here we are not considering the presence or absence of endosperm. Instead we are taking in consideration of another structure that is called as nucellus. What happened naturally? The third type of seed is called as the perispermic seed. Perispermic seed, naturally what happened with the development of embryo sac and the embryogeny, the nucellus is being completely digested or being completely consumed. But in some of the cases, nucellus remains persistent. Nucellus remains persistent as a residual structure. So if the seed is having the residual persistent nucellus, that is known as perisperm. What is perisperm? Perisperm is the residual persistent nucellus. Perisperm is what? If it is a question, what do you mean by perisperm? So perisperm is a residual persistent nucellus. So if the seed is having the residual persistent nucellus, then the seed is called as perispermic seed, because that residual nucellus is called as perisperm. The example for that is the black pepper and the beetroot. Okay, so two changes are now clear to you all. The first change is the zygote is being converted in form of embryo. The second change, the PE and primary endosperm nucleus is changed in form of the endosperm, and based on the presence or absence of the endosperm, albuminous, X-albuminous, and somewhere the perisperm is also present. So that is called as the perispermic seed. The third change what we have observed, the nucellus of embryo sac, nucellus of the female gametophyte is being digested, but sometime it is being persistent. Just now we have seen such as in case of the perispermic seed. So the black pepper and beet is the example for that. So is it clear to you all? Normally the nucellus is being digested, but sometime it may persistent, and whenever it is being persistent, so that time it is called as perisperm. You know very well. Now we have just cleared the concept about that. So now moving on to the next. Okay, so next structure after the nucellus, the integument of the ovule. The integument of the ovule is going to convert in form of the seed coat. The integument of the ovule is going to form the seed coat. You knows very well there are two integuments were present, the outer integument and the inner integument. So definitely the two integuments are there, so there will be two layers in the seed coat. Seed coat means what? The external covering of the seed is called as seed coat. So two integuments are there, so these two integuments are going to make the two different layers of the seed coat. So let us see what are those layers. The outer integument is going to form the external layer of the seed coat, that is called as testa. The outer layer of the integument is going to form the outer layer of the seed coat. Outer integument is going to form the outer layer of the seed coat, that is called as testa. Testa is considered as the hard and leathery coating, which ensures, why this is hard and leathery and the external region, why it is appeared on the external surface only. This ensures the survival of the seeds even on the harsh condition, even on the unfavorable condition. This increase the survival rate of the seed. The inner integument, the next layer of the integument, that is inner integument, is a thin layer, is going to form the thin layer inside the testa, that is called as tegument. So it is clear to you all. The seed coat itself is made of two layers, testa and tegument. Testa is the converted form of the outer integument, and tegument is the converted form of the inner integument. So now it is clear to you all. Now moving on to the next change. The next change is the micropyle. In the female gametophyte, in the ovule, the next structure is the micropyle. So micropyle is always present in form of the small pore. What is there? The micropyle is always present in form of the small pore in the seed. You can always find there at the bottom of the seed a shallow depression is present. That shallow depression is actually the representation of the micropyle. What is the importance of the micropyle, or what is the importance of that seed pore? We can say the seed pore. The seed pore is very much important, which in facilitation of absorption of oxygen and the water during the process of germination. Are you getting my point? What happens from that seed pore? The micropyle is present in form of a small pore or shallow depression there, called as the seed pore. What is the role of that seed pore? Seed pore is helpful in the entry of oxygen and water during the germination. Means what? This is very much useful there in the germination process only. Now next, the structure of seed. Actually, student, till now whatever we have discussed, the development of seed, that is what the structure of seed. We have seen the structure of seed. As if I am just going to give few points so you can understand well, and then you have to go with the structure of seed only. The seed is externally covered with a thick covering, that is called as seed coat. You knows very well. So whatever the structure of the seed is present inside that seed coat are considered as the kernel. Are you getting my point? All the structures present inside the seed coat is called as kernel. So clear? We have completed the complete development. Now we have understood that the external surface of the seed is covered with the external covering, that is called as the seed coat. So whatever the structures are present inside the seed coat, that is called as kernel. So whatever the structures are present inside the seed coat is called as kernel. So now, as per the structural point of view, the seed is considered of consists of the three parts. The first part is called as seed coat, second part is called as endosperm, and third part is itself called as the embryo, that is embryonal axis, from which the development of root and shoot system will occur. So seed coat we knows very well. The seed coat is the peripheral region developed from the integument, testa and tegument. Two layers are there. Testa is hard and leathery. Tegument is the simple thin layer. Endosperm, we have seen somewhere the seeds are having the endosperm present over there, and somewhere the seeds are not having endosperm. X albuminous, albuminous seeds, you can go with those description. Embryo is considered as the structure which is having the embryonal axis, epicotyle and hypocotyle, by which the root system and shoot system has to be developed. Now we are just going to see the diagram of a seed structure. These diagrams are very important. If you are able to see, I am just going to show the diagram of a dicot seed. Here in case of dicot seed, the outermost layer is called as testa. The inner layer is called as tegmen. So testa and tegmen both combine to form what? Seed coat. You know very well. So testa and tegmen combine to form seed coat. Inside that we have understood there in the development of seed only, because in dicot there is no endosperm present, so for giving the nutrition for the purpose of germination, the stored material is present in form of what? Cotyledons. So because they are dicot, so they will have the two cotyledons present over there. This is what the embryo. So three structures are there. Seed coat we have completed, endosperm that is in presence of in form of the cotyledon, because endosperm is now digested therein, and embryo is present in form of the embryonal axis. So this is what the embryonal axis, and in embryonal axis the upper portion is called as epicotyle, you knows very well, and the lower portion is called as hypocotyle. So epicotyle give rise the plumule, will terminate in plumule, and hypocotyle terminates in the radicle. So radicle will give rise the root system, and plumule is going to give rise the shoot system. So this is what the diagram of, or this is what the structure of an dicot seed. Now let us see the structure of another seed, that is once again the dicot plant, but it is having what? The albuminous seed. Remember the example of the castor, example of castor and papaya we have seen. So castor seed is dicot but having the albuminous nature. This diagram is being asked there in the NEET examination, so just take care of that. This diagram of the castor seed along with the monocot seed is being asked to find out which structure are being labeled over there. So this is what the labeling is there. So outer layer is making the seed coat, inside the endosperm is present, and cotyledon is also present thereon, and this is what the embryonal axis present herein. So embryonal axis, the bottommost region is hypocotyle. That was asked the question. This portion was labeled in a diagram and being asked what is the name of this structure. So this is hypocotyle, and here in the external view only the hypocotyle is there. This portion is considered as the root meristem, root tip, and this is what the shoot tip. So shoot tip or shoot meristem is going to give rise the shoot system. Root tip, you can understand well कि from here only the root has to be developed, and this is going to protect the root system only. So this is what the question has been asked, कि this structure is what? So A and B structure, like that. If C is there, D is there, so you have to go for the identification of this one. So let us move on to the structure of the monocot seed, or the seed of grass family. Many times the question has been asked, you just describe the structure of a grass family seed, or a grass seed, or a maize seed, or a wheat seed, or whatsoever is there. But remember, whatever the monocot plants are there, they will have the same kind of the structure. So listen, because they do not have the two cotyledons, so simple structure or straightforward structure is there. So the outer one layer is pericarp, which is combined with the testa and tegment, which is combined with the seed coat. So outermost layer is pericarp, considered as the is being found combined with the seed coat. So this one is clear to you all. The pericarp is the external most layer, which is combined or joined with the seed coat. Inside that the endosperm is present there, because the seeds are albuminous and with the persistent endosperm. This portion is representing the scutellum. Scutellum we'll see later on. The scutellum is actually the rudimentary cotyledon. In case of monocotyledon, scutellum is referred as the cotyledon. And here this is what the embryonal axis. This is complete embryonal axis. Here we are able to see the coleoptile. We are able to see the plumule. So coleoptile is a theca or thecus are covering around the plumule present. And here we are able to see the radicle and the coleorhiza. So coleorhiza is the structure which encircles the radicle. And one important thing that I have written here, aileron. So sometime endosperm is very much rich there in the protein content. So that time this is called as what? Aileron. So is this clear? Very simple, straightforward diagram is there, but very important question it is there. So pericarp, endosperm, scutellum, coleoptile, plumule, coleorhiza and the radicle, and this is what the embryonal axis. Once again all three diagrams. So this is what the testa and tegmen in case of the dicot seed, cotyledons, the embryonal axis, plumule, radicle, epicotyle, hypocotyle. They are in case of another dicot seed, but it is albuminous in nature, so it will persistent with the endosperm, the castor seed, the seed coat, endosperm, cotyledon, and this is what the embryonal axis. The final region is called as hypocotyle, that has been asked, that is why I have just drawn it separately. Now once again the monocot seed. So monocot seed, the pericarp, the fruit wall along with the seed coat, endosperm is present there, scutellum is present there, aileron layer is present, coleoptile, plumule, radicle, coleorhiza, all these structures are present there in the monocot seed. Okay. So after seeing the structure or the diagram of the monocot seed, remember these are very crucial point and repeatedly they are being asked there in the examination. Okay, what is scutellum? Scutellum is actually the form of cotyledon present there in the monocot seed. In monocot seed, cotyledon is known as scutellum. The embryonal axis of the monocot seed is having four structure: the radicle along with the coleorhiza. Coleorhiza is encircling the radicle. Plumule along with the coleoptile. So coleoptile is the structure which encircles the plumule. Coleorhiza is the structure which encircles the radicle. Aileron layer, very important question. This is a triploid structure, that is the protein rich endosperm. Whenever the aileron layer is there, so aileron layer is called as is considered as the protein rich endosperm. So now this is what the structure and the development of seed. Now we are moving toward the germination of seed, how the seed is being able to germinate. So let us see the seed germination. Normally after the development of seed, after the maturation of the seed may just get inactive to ensure their long time survival, because this is not necessary whenever they are just getting matured, so they may get the enough amount of the requirement or enough requirement to get germinate over. And if such conditions are there and if the seeds are not able to maintain their survival, so they may get fruitless. So that is what the process is not being beneficial. So that is why seeds are making a kind of adaptation, they are just getting inactive. But whenever the conditions are favorable for their germination, they are just getting activated. So conversion of inactive seed or inactive embryo inside the seed into the active embryo is called as the germination. What is germination? Germination is actually the conversion of inactive stage of seed into the active stage. What is inactive stage? Non-ability to germinate, and active stage is ability to germinate. So ability of the seed to produce the seedling in presence of the favorable conditions like moisture, oxygen and temperature. What is seed germination? The ability of seed to produce the seedlings in presence of the favorable conditions like moisture, oxygen and temperature. Are you getting my point? Just now I just made you clear that at the time of seed formation, this is not necessary the conditions should be favorable for the seed. So what happened? They are waiting till the conditions should get favorable. What are those conditions? Enough amount of the moisture, enough availability of oxygen, adequate temperature is required thereon. So whenever these conditions are getting available thereon, so seed is able to produce their own seedling. So that is what called as seed germination. So ability of seed to produce seedling is called as germination. So based on the basis of their process, how the seed germination occurs. So germination is of two types. First one is called as epigel germination. My dear child, EP means upside, GL means the ground. EP means upside, GL means ground. You know very well in the etymology we are able to differentiate, we are able to divide those terms, we are able to break those terms. So EP means upside, GL means ground. Germination. So EPGL germination, upside ground germination, whenever the germination process was observed or is observed over the surface of ground. So this process is very important. Remember, I am just going to provide you the key points only. Epigel germination is completely depending upon the growth of hypocotyle. Epigel germination is depending upon the growth of hypocotyle. Just opposite, EP to hypo. Remember, this is a kind of confusing statement. EP to hypo. Epigel germination is being is being stimulated by the growth of hypocotyle, not by epicotyle. So ep to hypo, and similarly hypo to ep will be there. So whenever epigl germination is there, so the growth of hypocotyle will be recorded, and due to the growth of hypocotyle, the cotyledons are being pushed over the surface of the soil. Are you getting my point? Due to the growth of hypocotyle, the cotyledons are pushed over the surface of soil. That is why this is called as what? Epigl germination. Rest of the parts remains inside the soil only. So are you getting my point? So when cotyledons remain inside the soil during the process of germination, that is called as what? Epigl germination. So due to the growth of hypocotyle, cotyledons pushed over the surface of soil. Example: capsula, castor, tamarind, etc. So ep to hypo. Now next type of the germination is called as hypogel germination. So this is vice versa to the next one, previous one, hypo to ep. So hypogel, hypo means below the soil, hypo means below, gel means soil. So once again we are just breaking: hypogel, hypo means below, gel means soil. So hypogel, below the soil germination. So whenever the hypogel germination is there, so hypo to ep. So due to the growth of epicotyle, what happened? Plumule will comes over the soil, but cotyledon will remain inside the soil. Are you getting my point? Due to the growth of epicotyle, what will happen? Plumule will comes over the soil, but cotyledon will remain inside the soil only, or will remain underground. So remember here, the types of germination is not based on the plumule and radicle. Instead, it is based on the location of cotyledon, where the cotyledons are present. So in hypogel, cotyledon remains inside the soil, but plumules comes out, and due to the growth of epicotyle. And in case of epigel, the cotyledon comes out due to the growth of hypocotyle. So example of the hypogel germination: most of the monocots are having the hypogel germination, but similarly some of the dicots are also having, like gram, peanut, etc. One very important, very rare case, a specific case of germination we are just going to discuss, that is called as vivipary. Vivipary is the property of the mangrove vegetation. Mangrove vegetation, example of mangrove vegetation is the Rhizophora and Caryopsis. Mangrove vegetation are specific kind of vegetation, those are present there in the saline condition, they are on the bank of the oceans. So mangrove vegetation are the specific vegetations present on the bank of the ocean. In the saline condition, the vegetation grows in the salinity, so they are not having enough amount of the air, oxygen and everything, so that they could able to germinate properly. So that's why they have adopted a better way of germination. The seeds are being germinated inside the fruit itself whenever they are just being developed, or they are being growing. So what is there in viviparous? Viviparous, the seeds are germinated inside the fruit only while they are attached to the parent plant. Are you getting my point? The seeds are germinate inside the fruits while they are attached to the parent plant. That is why this process is called as what? In situ germination also. The seed is germinated there only, in the parent plant only, inside the fruit only. That is why it vivipary. Vivipary means what? The development of the young one inside the mother's body. And here, because they are developed in the parent's body, inside the fruit only, where they are being developed, so that is why this is called as in situ germination only. And why they have just such kind of the development? Because the non-availability of the proper condition of the germination. One more important point that we have to see, that is seed dormancy. Seed dormancy, we can say the seed is able to germinate, then seed germination, but sometimes the seed is not able to germinate themselves, seed fails to germinate themselves. That condition or that duration is called as seed dormancy. Seed dormancy is the state of inactive embryo. The seed dormancy is the state of inactive embryo. The seed is not able to germinate. Are you getting my point? The seed dormancy, dormancy का मतलब है सो जाना, dormancy is slept. That is not inactive. That is not we cannot, the seed is getting died, but seed is temporarily not able to germinate. So state of inactive embryo is called as seed dormancy. कुछ ऐसा है कि आप सो रहे हैं और सोते समय आपके पास में सारी मेटाबॉलिज्म चल रही है बट यू आर नॉट एबल टू wake up. Similarly, the seed is having every metabolism but the metabolic rate is very much slow. They are not able to germinate because the conditions are not favorable. But whenever the conditions are getting favorable, the seed is able to germinate. Then the germination will be there. So what are those conditions on which the seed dormancy occurs? The dryness, basically the dryness induces the reduction in the water and the slow metabolism. What is the identification of seed dormancy? The dryness induced reduction in the water and the slow metabolism. Due to the dryness, there is less amount of the water is available in the soil and that will going to promote the slow metabolism also. So this is what the seed dormancy. Students, now we are going to discuss what are the factors which are going to regulate the germination of the seeds. So factors which are going to regulate the germination of seeds, the first factor is moisture. Basically 3 major factors are there. The major factors: air, water and temperature. So water in form of the moisture. The dormant seed or inactive seed is having around 10 to 15% of the water. Dormant seed or inactive seed is having 10 to 15% of the water. But for activation of the seed, means what, to perform the active metabolism of the seed, they require the 75 to 90% of the water. What is the meaning of that? Normally the dormant seed or inactive seed is having what, 10 to 15% of the water, but for converting itself in form of in form of an active seed, to start an active metabolism, they demand at least 75 to 90% of water. So whenever the adequate amount of water is present, so they are able to germinate itself, otherwise they are fails to germinate themselves. Next one, oxygen, air. Oxygen is very important component, you know very well, for the purpose of germination also. Even though seed is having the cotyledons or endosperm present over there, so these cotyledons or endosperm is not able to give the energy for the purpose of germination. So for the purpose of germination, whatever the energy is required, that energy will be provided by the oxidation of those organic material present inside the seed. So that oxidation will be performed by the presence of adequate amount of oxygen. If the oxygen is not available there, so how will they get the energy for the purpose of germination? The next one, the temperature is playing a key role there in the germination of seeds. Here the seed is not able to germinate when the temperature falls in between 0 to 5 degrees centigrade, or the temperature is above 45 degrees centigrade. The seed is not able to germinate the temperature in between 0 to 5 degrees centigrade or above the 45 degrees centigrade. The optimal range of the temperature for the germination of the seed is 20 to 25 degrees centigrade. The optimal temperature range for the germination of the seed is 20 to 25 degrees centigrade. So why the seed is failed to germinate in between those temperature in 0 to 5 degrees centigrade or above the 45 degrees centigrade? My dear student, in the previous classes you have gone through the concept of enzyme physiology, enzyme mechanism. In enzyme physiology or enzyme mechanism, we knows very well the enzymes are very playing a key role there in the metabolic processes. If the enzyme is not present there in adequate quantity or enzyme is not active, so the metabolic rate gets lower than down, and the temperature is the key factor for influencing the enzyme activity. If you have remember, the enzymes get inactive at the range of 0 to 5 degrees centigrade. So that is why the 0 to 5 degrees centigrade most of the seeds are not able to germinate, and enzymes are getting denatured over the 45 degrees centigrade. So this is what the enzymes are not able to perform their activities, and the seeds are not able to germinate over the 45 degrees centigrade. Lastly, the nutrition. So nutrition is present in form of either cotyledon in dicot seed and in form of endosperm in monocot seed for the purpose of germination. The seed viability is the last concept for the seed today. The seed viability means viability, the living capacity of germination, the time period for which the seed can be considered as the active and having the capacity to germinate itself. So the seed viability means the period for which seed is able to germinate itself. The period for which the seed is able to germinate itself is called as seed viability. What is seed viability? It is the period for which seed is able to germinate. So that is highly variable in different type of the seeds. We cannot say the seed viability or the period of seed germination for every seed is common or constant, but it is reported from few months, few days, few months, few weeks to many of the years also. The most oldest record of the viability of the seed are being recorded from the oldest seed, that is lupin of the Lupinus arcticus. The seed is having 10,000 year of the dormancy. Are you getting my point? So after 10,000 year of the dormancy, it is able to germinate itself. So oldest seed which is recorded for the germination, the oldest seed which is recorded for the dormancy is the lupin seed, Lupinus arcticus. 10,000 year of the dormancy has been reported. The next seed which is date palm, Phoenix dactylifera, 2000 year of the seed dormancy. 2000 year of the seed dormancy. So oldest seed is lupin, 10,000 year of the seed dormancy has been reported. The next seed that is reported for the seed dormancy, longest seed dormancy, is date palm, Phoenix dactylifera, 2000 year of the seed dormancy. Next concept that I am going to give you, that in the box only it is written, so means if it is written in the box, so ultimately it is going to be very important. Some of the tests are being performed to check the viability of the seed, means what, the seed is able to germinate or not. So if the seed is not able to germinate, so why to give it to the farmer, so that the process of farming is getting futile. So to avoid such kind of the practices, the seed viability is being tested. So for which or how the seed viability is being tested, so for checking the seed viability, which component is being used, which chemical is used, TTC. TTC is 2,3,5 triphenyl tetrazolium chloride. The seed viability is tested by TTC 2,3,5 triphenyl tetrazolium chloride. How the seed viability is tested? By TTC, application of TTC on the seed. And what is TTC? 2,3,5 triphenyl tetrazolium chloride. After treating the seed with the TTC, the embryonal axis of the seed, if it is in the living condition, become pink or turn in the pink condition, pink color in the TTC solution. When the seed is being treated with the TTC, if the seed is having ability to germinate, the embryonal axis will convert in form of the pink color, will show the pink color thereon. So this is all about the seeds and everything. Now next topic we'll take, that is the last one of the post fertilization changes, that is the development of fruit. So I think now everything is clear to you all. Today we have discussed what, the development of seed along with the structure of seed in the monocot, dicot, and some of the seeds which are very specific like the castor seed, the diagram, then the seed germination, and the factors which are being affected to the seed germination. So just go through with the topic, and we'll start the next one in the next lecture only. Till the time, thank you.